Hello, my name's Jack Foreman, and for the extra credit assignment, I am demonstrating amino acids. So in my hand, I have a single amino acid with an R group here, a carboxylic group, and an amine group. This comes together with another chain of amino acids, and in a condensation reaction, the amine group loses a hydrogen, and the carboxylic group loses an OH, and they form a chain of amino acids. And over here, we get a water group, making it a condensation reaction. So in this structure, we have several different elements to it. So right here, we demonstrate the OH bond, or the carbon-oxygen double bond. And in this group, we demonstrate how this structure has resonance, making this whole structure planar, allowing no rotation. This means that the only rotation, or the primary rotation, is right here and here, the angle psi and the angle phi. Both these allow for the construction of secondary structures, which are shown here, primarily the alpha helix and the beta sheet. The alpha helix is shown by this ribbon structure that forms a helix, and there is a carboxylic end, like so, and a amine end, and they line up in order, and at one end you have the amine end, and on one end you have the carboxylic end, and these form hydrogen bonds between each other, creating this solid structure. In beta sheets, we have the same idea where we have a carboxylic end and an amine end, but they can also be parallel, so either the amine end is on the same side, amine ends are on the same side, or it's a amine and a carboxylic end on the same side. And these form two different structures, a parallel and an anti-parallel. Parallel, you see these kind of groups of hydrogen bonding, where they just have two parallel hydrogen bonds shown by this hydrogen bonding to a oxygen over here in a hydrogen bond. Whereas parallel makes this zigzag shape with the hydrogen bonds, and that's one way to tell them apart. These sheets come in big strands, and they can, same with these, and they can have rotations, or B-turns. B-turns are typically seen by amino acids, particularly glycine and proline, because the R groups on each of these are very significant, and they allow, glycine has a small R group, which allows it to rotate better, and proline has a cyclic structure that also allows it to rotate better, forming these turns.